Yeah, it will be a long night. All right, so we left off last time talking about enzymes and how enzymes can be inhibited either directly through the active site. So that's where the substrate's going to bind to that active site. And that can cause the enzyme to not be able to work, or it can work at a distant site that is not part of the active site. But in doing so, in binding to the that other site, it does inhibit. It's called a non-competitive inhibitor. And what it does is it still affects the active site, but it affects it by actually changing the site of changing the shape of the active site. So we saw that here, where the non-competitive changes the shape of that active site, and now the substrate doesn't really fit nearly as well as it was as it did before. So those are all examples of inhibition that are not normal. That could be toxins, poisons, things in the environment that get into our bodies and then negatively affect the functioning of our enzymes. 8.5 talks about how enzyme activity can be regulated through something called an allosteric site. And that's called allosteric regulation. And that is not the active site. It's a site that is distant from the active site. But it has similar, well, it really has the same effects as a non-competitive inhibitor would, in that it's going to change the shape of the active site. But an allosteric regulator could work either positively to keep an enzyme turned on and make it more functional, more easy to bind to a substrate, or it could turn it off to make it harder for a substrate to bind to a substance. And this is done in order to make sure that enzymes are only being made and they're only being used when they're needed. Because for many different reasons, we don't want all of our enzymes being activated all at once. Some of those can be because of the fact that there's energy being used. These enzymes are going to break down things that don't necessarily need to be broken down, and that could require energy in doing that. We don't want to waste energy. It could be that it could be actually catastrophic. Maybe that particular breakdown, that particular reaction would actually make a substance be broken down that we really want to keep whole, that we don't want to digest it or break it down. Most allosterically regulated enzymes are made from polypeptide subunits, each with their active site. The enzyme complex has an active and an inactive form. And that has to do with that active site. Either that active site shape is the active site. I mean, it's the right shape because it's active. Or it has a shape that the substrate can no longer fit in, and that's the inactive form. There are activators and there are inhibitors. But again, in this sense, when we're using activators or inhibitors, but when we're talking about inhibitors in this case, we're not talking about the non-competitive inhibitor that is harmful, that is a poison, it's a toxin. These are regulators. These are molecules whose job it is to regulate the enzyme's function, making sure it's turned on when it should be and making sure it's turned off when it should be. This is an example of this. It's just a very basic one where you've got an enzyme that actually has 4 subunits. And you've got this little green molecule here. And that little green molecule fits in this little wedge. And in doing so, it keeps that active site open, keeps it in that kind of U shape. Make that a little bit bigger here. I think I make it smaller, which is weird, but yes, very strange. Let me clear that out. Yeah, so I wanted it cleared. There we go. So that little thing wedges in there, and that keeps this site open so that substrate molecules can bind. In the second picture, we have inhibitors. I use a different color since that's red. Now I've got an inhibitor, and that wedges in there, and the way it wedges in there, it closes up that active site. It's no longer U-shaped. Because it's no longer U-shaped, the substrate will come along and it will, try and get in there. So if this was my substrate's like going to fit. It's like, yay, I'm happy. And in this case, that U-shaped substrate tries to get in there and boom, rejected. Sorry. In that case, that molecule will not be broken down. It is common in metabolism for there to be a series of reactions that occurs. And we talked about this at the beginning of the chapter, where you would have a molecule that becomes another molecule, that becomes a third, that becomes a fourth, maybe even a fifth. I mean, some of these are really long. Like in biochemistry, you learn about all of the reactions in cell respiration and in photosynthesis. And I think in glycolysis, the first stage of cell respiration alone, there's 16 reactions. So this is just a small chunk of what could really happen. But what happens is if you make enough of E will build up, its concentration will build up enough that it will come back to A. Or actually, let me, I shouldn't have cleared the whole screen. My bad. We'll just go to C this time. Actually, let's just go on. What the heck? And it will feed back here to that enzyme. So each one of these has an enzyme associated with. A to B would have an enzyme, B to C, C to D, and D to E. And what it will do, is if that E concentration is high enough, it shuts it down by doing that allosteric regulation. It changes the shape of the active site, and now it's not going to work anymore. And that's actually shown on this next page. You do not have to memorize the steps in this. You're starting out with a particular substrate called threonine. Threonine's an amino acid. Just so you know, there's 21 different amino acids. Threonine's one of them. And threonine can be changed through a series of reactions. So I've got an enzyme here, I've got an enzyme here, here, and here. And eventually it gets changed into this end product, which is another amino acid called isoleucine. And again, you don't need to know the name of the amino acids. It's basically you're just going from one amino acid and you're making it into a different amino acid. When enough of that isoleucine has been made, that is the product, the end result of that last reaction, it's going to come back to that purple. Remember, proteins are usually purple. In this case, it's a enzyme that we're looking at. It's going to change that shape. This is what the active site's supposed to look like. And this is what it looks like when the threonine comes in here and it changes the shape. The shape changes. And because the shape has changed, now this beginning molecule cannot bind to its enzyme, and none of these reactions will proceed. All of them will be shut down. So that's an energy saver. Because each one of these, one thing that's not being shown in here is that probably at least some of these are going to require some of our, what is our energy molecule? ATP. So some of these are going to require ATP. And so if we shut them down, if we shut them all down, then we conserve energy. So if there's a case where, you've made enough, we'll shut it down and keep that from wasting a lot of energy and ending up with a surplus of that amino acid that we really don't need. Enzymes have different locations within the cell. Oftentimes they are in structures that are close to the metabolic pathways that they are going to facilitate. And sometimes that's actually in the cell membrane. They'll actually be embedded in the cell membrane. We'll talk about that when we get to cell communication. They could also reside in a particular organelle. When we get to cell respiration in just a minute, that will be the mitochondria. We'll talk about how there are enzymes embedded in the cell membrane, in the membrane of the mitochondria that are helping facilitate the production of ATP. So there's all sorts of places that you can find enzymes, but they're usually located close to where they are going to be needed, which makes sense. All right. That is all of chapter 8. Any questions on that? All right. Now, this looks like a badly redacted, well, it is a badly redacted slide. For whatever reason, when I made these slides a long time ago, I came across this weird glitch where it would, if I copied the slide from the beginning of the slideshow, if I copied the slide from another slideshow and I put it in here, the chapter 8 one, so chapter 8, this guy, it instead would default back to the first slide in the slide deck, and it would show chapter 8. No matter what I did, it defaulted back to the first slide in the deck. So that's why it's typed in here, chapter 9, cell respiration, and why this has been deleted out, because it actually talked about chapter 8. This is about chapter 9. I couldn't figure out how to get it to be okay with me wanting to go to the next chapter. Even though I copied the slide and I pasted it in, it just wouldn't do it. But this is going to be on cell respiration. So now we're going to talk about, now that we know a little bit about energy and how enzymes speed up reactions and can be turned on and turned off. Now we want to talk about how that energy is put to use. In order for living things to live, they have to get energy from the outside world. And that could be through photosynthesis if you're a plant, or if you are a non-autotrophic organism, not a plant, in other words, you can do that through this thing called cell respiration. So animals can obtain energy to do work by feeding on either animals or by photosynthetic organisms. And we're going to talk about how they actually take that energy from plants and from animals and convert it into a form of energy that they can use. Remember that energy is going to flow from our sun, and it's going to go to plants, and some of that energy as it flows is going to be lost, as we talked about this in the last chapter. We talked about it's going to be lost as heat. And that heat goes away. It doesn't come back. We have to constantly have a source of energy, that being our sun, because we lose some energy every time we change energy from one form to another. So when we change sunlight into chemical energy through photosynthesis, when we eat our food, the energy that is in the bonds of those molecules is not going to be perfectly used. Some of it's going to be lost as heat. Okay, and like I said before, everyone poops. Some of it's also lost through digestion, so we don't use all that. But a lot of the structural components we do reuse, in terms of the elements. We reuse those elements over and over again. The thing is, to put them together, we have to use energy to recombine them in a way that is helpful for us. And that's shown actually on this next photo, which is kind of a simple one, but it is important. It illustrates what's going on in our ecosystem. We're getting we're getting light from the sun. So we're getting this light energy in here. And it's being taken in then by our chloroplasts that are within our plants. And they are making organic molecules that they will use to grow and divide and grow again, as well as for our use, right? We eat plants, and we're going to benefit from them as well. After photosynthesis, we make those yummy molecules that then have to be broken down in cell respiration. And that happens partly in the mitochondria, but not completely. I know it says the mitochondria here, but not all cell respiration takes place in the mitochondria. We'll talk about that. And then from there, that mitochondria and cell respiration produce ATP. They produce our energy that then we can use to do whatever we need to in our body. Moving things around, breathing, thinking, dividing cells, whatever it happens to be. That process takes in, the process of photosynthesis is going to take in carbon dioxide, and it's going to release out oxygen. Okay, and that oxygen that plants are going to give off is the oxygen that we breathe. And that's going to be important for cell respiration. That's kind of an overview of what we're going to talk about in the next couple of chapters. Okay. Nope, my son is still there. Remember, we talked about catabolic pathways. And catabolic are pathways where you're taking larger molecules and breaking them down into smaller ones. And when you do that, you are going to release energy. The release of energy from those complex molecules is going to, in large part, be from the movement of electrons from one molecule to another. Back in the day, we talked about electrons and talked about how they're moving, they're orbiting around, they have energy associated with them, right? If I take a ball on a string and I'm swinging that ball on that string around in a circle, there's energy associated with that, right? If I went up to somebody with that ball on the string and I hit them in the head, it would hurt, right? Because there's energy associated with it. Anybody ever play tetherball? right? Right? Anybody ever get smacked in the head playing tetherball? Yeah. Okay. So there's energy there. Okay. And the electrons is that ball. So we can move that ball from one location to another, and in doing so, we're moving that energy with it. And that's going to be pretty key in our cell respiration. Cell respiration is a catabolic pathway. It is a series of catabolic pathways, I should say. There are a few of the reactions within cell respiration that do require energy. But for the most part, it's going to be energy releasing, or ATP producing, I should say. So the breakdown of organic molecules, like our food is organic, is exergonic. Okay, catabolic again is exergonic. It's going to relieve, or it's going to give off energy. When we talk about respiration, we can talk about fermentation, and we can talk about aerobic respiration. And I don't want to dwell on these bits too much right now, because we're going to go over them in more detail, and I think it will make a lot more sense than just giving you the definitions right now. If we just go over them as we go along, I think it will make more sense. So when we talk about cellular respiration, we are not talking about just breathing. Breathing is a very minor part of respiration, of cell respiration. And it's referring to what we call anaerobic and aerobic respiration, okay? Which means that part of it is going to use air. What does that refer to? Aerobic oxygen. So these are processes that require oxygen. And then you have the anaerobic. How about them? Do they require oxygen? No. No, they do not. So O2, no. Okay. So cell respiration is a combination of those two things together, but we often refer to it kind of synonymously as just aerobic respiration. And our bodies, when our body makes ATP, when our body makes energy, it would prefer to work with this molecule called glucose. So a lot of the things that we eat get converted into glucose, or parts of glucose, as they go through this cell respiration process. In here, for this class, we are going to just talk about how glucose gets broken down. We're not going to look very much, we'll spend a little bit of time, but not much, on fats and proteins. But keep in mind that it isn't just glucose that's getting used in cell respiration. It's every part of your food. Even the nucleic acids, the DNA, even that is getting broken down and being used. But the proteins, the fats, all of those things are being used to produce ATP. We're just going to talk about one carbohydrate, and that carbohydrate is going to be glucose. Okay. All right. So the equation, and you should know this formula, is C6H12O6. That is what? Glucose. Yeah, that's glucose. And oxygen is going to give us carbon dioxide, water, and energy in the form of both ATP as well as heat. So one of the things that happens when you look at this process is you actually find that a lot of your energy is lost as heat. We're not that efficient at digesting our food. A lot of it gets wasted. But the bonus is it keeps us warm. Come on. Okay, so back to the electron thing. When we're talking about electrons, electrons can be moved. They can be passed from one molecule to another. And when they are passed from one molecule to another, let me start with this. What is the charge on an electron? Negative, right? Okay. So whenever something takes in an electron, actually, let me go to the next page. I think whenever something takes in an electron, we call that reduction. It's like you're becoming more negative. You're taking in an electron, you're becoming more negative, you're becoming reduced. But it's always in pairs. If somebody gains, somebody else has to lose, right? Play football, there's one winner and there's one loser. In electron transfer, you have one winner and you have one loser. Okay. Well, I don't know if it's loser, but one person or one, I got to stop that. One molecule is going to lose its electrons or lose an electron. And we call that oxidation. The one that loses the electron has become oxidized. Sometimes these are referred to as redox reactions because reduction and oxidation, so redox. But they always happen in pairs. If one gains, the other one loses. So in the case of sodium and chloride, you put those two together and you get sodium and chloride ions. And this guy here, the sodium's become what? It's lost an electron. Oxidized. And this little guy, he gained an electron, so he's become reduced. Okay? Reduced and oxidized. That happens, the movement of electrons in this process happens often and is really relevant to the process because without the movement of these electrons, you would not get nearly as much energy. And we'll talk about why that is or how that is as we go along. We probably will not do that today, though, because it is a long process. Okay. During cell respiration, the fuel, like glucose, again, we're going to use glucose, but it could be fats, it could be lipids, his lipids, or his fats. We could use proteins, could use nucleic acids. Glucose becomes oxidized. So if glucose has become oxidized, what is it losing? Electrons. In the very, 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 very end, oxygen is going to pick up those electrons. And we say, when that happens, we say that it has become reduced because it's going to take in those electrons at the very end. Organic molecules have a lot of hydrogen. We talk about C6H12O6, a lot of hydrogens. And along with those hydrogens are a lot of electrons, a lot of high energy electrons. The energy associated with the hydrogen ions is released when it gets transferred from one molecule to another and eventually to oxygen. If you think about, I think there might be a picture of this later. If you think about stairs and you think of the ball at the top of the stairs, the ball ball's at the top of the stairs. It's going to roll down those stairs. Those are steep stairs until it gets to the bottom. And once it's at the bottom, it's lost a lot of its energy. Right? There's a lot of energy that's like, can't really use that to do any work anymore. Those electrons are spent. And so it is the oxygen that's going to pick up those electrons when they're done and do something with them. Get rid of them, actually. In cell respiration, glucose as well as other food molecules are going to be broken down in a series of reactions. So it really is a stepwise transfer of energy. So those things happen in a stepwise fashion. Got a little bit of energy released, a little bit more. In order to move an electron from one place to another, it isn't always given directly to the molecule it's not directly given to oxygen. It has to be passed from where it is being given from the things becoming oxidized. It gets moved to this molecule called NAD. And NAD is like a taxi. A taxi can have a fare or it cannot have a fare. NAD plus is a taxi without a fare. It is a molecule without electrons. When electrons are released, those electrons will be passed to that NAD and they will become a reduced form because now they're taking in electrons and they become NADH. What are they also taking in besides an electron? A couple electrons. What's on that word that wasn't there before? H. They're also taking in H's, hydrogens, as well. NADH will literally move electrons from where they're captured from an oxidized molecule to the site of where ATP can be made. So when it says NADH, the reduced form of NAD plus represents stored energy that is tapped to synthesize ATP. That's what I'm saying. The NADH takes that electron from wherever it was made or not released, not made, and it will move it to the location where it can be used to synthesize ATP. I think this next picture shows you that graphic. There's my stir step. It shows you what NAD plus looks like. You don't need to know what NAD plus looks like. Really the only thing you need to know here is you have NAD plus, and NAD plus is going to gain a couple of electrons and protons. Actually clear that. NAD plus is going to gain a couple of electrons and a proton.
I think it's frozen.
Oh, weird.
Oh, there.
Wow, there's some lag going on. So it's going from NAD plus to it's adding from the food that we eat. It's going to take in a couple of electrons and a proton to become NADH. So is this oxidized or reduced? This one. This is oxidized. And this is reduced. Okay, that's my reduced form. It's now taken in electrons. It's gotten more negative charge. And it's NADH. You don't have to remember the name nicotinamide. NAD is fine. NADH passes these electrons that it's carrying, these electrons it's shuttling, it's taxiing, to an area of cell respiration called the electron transport. And as it does that, it's going to generate some ATP. This is believe it or not, an overview. Because we're going to talk more about this. We kind of have to set up what reduction and oxidation are before we can start talking about the actual stages of cell respiration. So all these things, electron transport chain, sometimes I call it ETC. ETC will happen down the road. Probably not in the next lecture, but the one after that. And the reason this happens, the reason it's step by step, again, like going down stair steps, is because if we did it all at once, that release of energy would be explosive. And really not beneficial. Probably pretty disastrous to our cells. So we want to release a little bit of energy in a step-by-step fashion. So release a little bit, then a little bit more, then a little bit more. And that way we can control the reactions and we can use them to do what we need them to do and not have to worry about things like, a catastrophic release of energy that might cause the cell to completely burst. It might cause all the cells around it to completely burst. So you have to have that step-by-step reaction. Okay, so last thing I'll talk about, and then we will end there. So those step-by-step reactions occur in a series of 3 stages. And those 3 stages are listed here. And this is what we will work on next time. Glycolysis, citric acid cycle, and, there's a fun word, oxidative phosphorylation. That's a big one. Let me ask you, phosphorylation, what do we talk about? What does that mean? We talked about that before. Oh, it's talking about adding on a phosphate group. When you add on a phosphate, you change the shape and you change its activation, whether or not it's on or off. So that's the phosphorylation process. ATP, most ATP is made in oxidative phosphorylation. We'll talk more about what that means, but I am going to wait and do that next time. So that is all for